Thanks for the nice invitation to join this nice and warm workshop. So I'm the only, I guess, non-English native speaker, so I might make some mistakes when I'm presenting. So I'm Bruno Guigas. I'm working in the Department of Parasitology in Leiden University Medical Centre in the Netherlands. And what I'm going to present you today is some of our work on the effect of helminths infections in the context of metabolic disorder. So I just realized that I might have had a question mark to my title, which is protective effect of helminths against metabolic disorders from worms to molecules. I hope I'll manage.
Is it moving forward? Just on the keyboard, is it?
On the keyboards there, yeah. Okay, sorry. So here, as an introduction, I just put this very well-known scheme showing that there is, over the last 50 years, there was a continuous decrease in the incidence of infectious disease, and this is associated with a concomitant rise in immune disorders, such as asthma or type 2 diabetes, so autoimmune disorders, inflammatory disease. But to this scheme, actually, you can add also the fact that in some of the westernized countries, there is also a complete eradication of some of the parasites, namely some helminths. And to the other side of these figures, you can also add the fact that there are continuous rise in the in the incidence of chronic metabolic disease. So if you, in these pictures from the World Health Organization, you can see that this is the human death throughout the 20th centuries, and you can clearly see that on the right side, most of the most important contributor to this worldwide death are actually infectious disease and non-communicable disease. But if you look a little bit more during the last decade, and that was introduced by the first speaker during the previous talk, there is a continuous trend for decrease in infectious disease, and this is associated with a rise in non-communicable disease throughout the world. And the contribution to the death from non-communicable disease is mostly due to an increase in cardiovascular disease, and to a less extent also to an increase in metabolic disease, such as type 2 diabetes. This is an example on how chronic disorders might develop differently depending on the degree of epidemiological transitions. So on this scheme, you can see that during age, there is an increase in the percentage of people suffering from hypertension, and that this increase, this age-related increase, is very different if you consider the location and the socio-economic states of the people that are developing hypertension. So what you can see in the yellow curve, for example, are people from the Netherlands. So they are developing high incidence of hypertension throughout life. But if you compare it, so they are living in the very urban areas. But if you compare that to people that are living in very rural environments, such as people in Flores Island in Indonesia, that's the green curve, you can see that they are developing much less hypertension throughout life. And this is also associated with high endemicity for helminth. So, and this map is also quite amazing, showing that the development of inflammatory disease worldwide, in fact, appears in countries where the helminths were not so much present. And if we come back to the chronic metabolic disease, such as type 2 diabetes and obesity, the first very important paper linking infections, helminth infections, and the regulation of whole body metabolic homeostasis was this landmark paper published by the group of Richard Loxley in Science 3 years ago, showing that in rodents, infection with a short-lived rodent nematodes called Nipostrongylus brasiliensis, in fact, improved the glucose tolerance and the insulin sensitivity of obese animals. And this is associated with some change in immune cells infiltration in adipose tissue. So, as I mentioned previously, there is a tremendous increase in metabolic disorder, especially in our westernized countries, but it's also the case in emerging countries. And well, if we go a little bit at the molecular level, this rise in metabolic disorders is associated with a kind of chronic low-grade inflammations. And these chronic low-grade inflammations is due to, well, in short, to infiltration of pro-inflammatory immune cells in metabolic tissue. This is an example in adipose tissue, where there are a bunch of immune cells that are colonizing your adipose tissue. And usually in lean persons, these immune cells are rather keeping a kind of anti-inflammatory state. So, eosinophils, macrophage polarized on M2 like macrophage, Th2 cells. And when you become obese, there is an increased infiltration in pro-inflammatory macrophage called M1 classically activated macrophage, and also an increase in Th1 cells that are producing cytokines that promote insulin resistance in your tissue. And interestingly, helminths are the well-known inducer of Th2 response, and they are promoting, in fact, immune cells that are, let's say, beneficial, the kind of immune cells that you can find in lean tissue like eosinophils and Th2, and they are also promoting polarization of macrophage toward a M2 phenotype. So just in brief, and that was previously described also by some previous speakers, helminths infections trigger some immune response, and the most important things you have to keep in mind is that helminths, after inducing a first acute Th1 response, they're promoting a Th2 response, which is controls when the infection becomes chronic, and this is mostly due to some molecules that are released by the eggs when produced by the worms. So I'm not going to describe this, but in the lab, in our group, we have described that some specific molecules derived from the worms can trigger the development of Th2 immune response. We have a model in our group where we infect a mouse, either put on a high or low-fat diet to induce obesity and insulin resistance, with helminths, which is called Schistosoma mansoni. And we have shown that when you infect mice, obese mice, so that's the figure on the right, when you infect obese mice with Schistosoma mansoni, you can clearly see that their body weight decrease over the time of infections. And this does not happen when these mice are on low-fat diets. In parallel to this, they also improve their insulin sensitivity. I'm not going to describe that in details, but they become more glucose tolerant, and they become more insulin sensitive at the whole body level. And this is associated with change in immune cell composition of the adipose tissue. So we measure by flow cytometry the composition of the immune cells in the adipose tissue. And you do remember that helminths trigger eosinophilia. And what we have first measured is the eosinophil content in the adipose tissue. And we found that the high-fat diet, so when you put your mice on high-fat diet, does not change so much the eosinophilia in the adipose tissue. But when you infect the animals, there is a very significant increase in eosinophils, whatever the kind of diet you give to the animals, whether they are lean or obese. And this happens in many adipose tissue in the body of these animals. So basically, this is gonadal, so just on top of the gonads, but also the visceral adipose tissue. It's exactly the same story. And we also determine the amounts of anti-inflammatory and pro-inflammatory macrophage in this adipose tissue. And these are called M1 and M2 macrophages. So the M1 are the classically activated macrophages, so the pro-inflammatory, and the M2 are the anti-inflammatory, alternatively activated macrophages. And what you can see is that when you put your animal on a high diet, there are an increase in the pro-inflammatory macrophage in this adipose tissue. And the M2 macrophages are not so much affected. What's happened when you infect your animal with helminth? There is not so much effect on the M1 macrophages, but there is a huge increase in the M2 macrophages. And so if you plot the ratio on M2 on M1, you have a very, very significant increase in the M2 macrophages in the adipose tissue. And this is true for all the fat pads we have investigated. And interestingly, this is also associated with an increased expression of some Th2 cytokine, especially IL-4, and also IL-5 and IL-13 that are known to be produced by ELC2s. So I will just skip this. So far, we have shown that S-Mansoni helminths improve whole body insulin action and also insulin sensitivity in the adipose tissue. And this is associated with an increase in eosinophils and alternatively activated M2 macrophages. And this is probably due to increase in ELC2 by the infections. So, but we are interested by to see whether if we administer the molecules that are produced by the worms or present in the eggs of these worms, we can mimic this phenotype. So what we have done, we have put first these animals on high-fat diet. They are becoming obese, and then we inject them during 4 weeks, every 3 days, with a mixture of molecules that are present in the eggs of this helminth. And we measure their glucose tolerance. So it's basically this kind of curve that I can describe. So animals on low-fat diet, you inject glucose to the animals, and then you follow the blood glucose level. And in lean animals, that's the open square, you can see that the glucose level goes back quite rapidly to the basal level. And the area under the curve reflects their glucose tolerance. If you put animals on high-fat diet, they become insulin resistant and glucose intolerant, and therefore their glucose excursion curves is very significantly increased. If you inject animals with this mixture of antigens, what's happened? Their glucose tolerance is partly restored. So meaning that these mixtures of antigens is beneficial in the context of metabolic disorders. And once again, we looked at on the composition of the immune cells in the adipose tissue, and once again, we found a very significant increase in eosinophils. We found a very strong increase in alternatively activated anti-inflammatory M2 macrophages, meaning that the ratio on M1 and M2 is rather toward an anti-inflammatory profile. And we also found a very significant increase in Th2 cells that are secreting EL4 and EL13. So actually, we have shown that SEA, the mixture of antigens, like the chronic helminth infections, are beneficial for whole body insulin sensitivity and glucose tolerance. The question, we don't know actually what's happened on other metabolic organs. We are studying that at the moment. But the second thing we don't know is whether it is only due to this change in immune cell infiltration in metabolic tissue or to direct effect of some of the antigens on the metabolic tissue. So, and there are only 2 slides just to show you that one of the arms we are considering is that some of these molecules can directly target metabolic tissue like adipose tissue, so adipocytes, hepatocytes, or skeletal muscle cells, and therefore trigger some signaling pathways that improve some of the metabolic processes that are impaired by metabolic disorders. So I will just show you one slide on hepatocytes. So what we have done, we have isolated hepatocytes from the liver of a mouse, and we have first looked whether some of these molecules from the helminths can bind and being internalized by the hepatocytes. And this is actually what you can see here. We have labeled this mix of antigens, and you can find these antigens inside the hepatocytes. And we have done that also with single molecules that are purified from this mix, and also found that some of these single molecules can enter inside the hepatocytes. And the liver is the main organs that regulate glucose homeostasis in the body, so they are producing glucose and taking up glucose and store glucose in form of glycogens. And when you're in the fasted state, so after your glucose is producing, your liver is producing glucose by gluconeogenesis, and we have looked on these metabolic processes. So to make a long story short, we have looked on some of the key genes that are controlling this process, and we found that that's the mixture of the antigens from the worms or some single molecules extracted from these mixtures, like omega-1, which is the glycoprotein we have isolated in our group and show that and that is potent for inducing Th2 response. We are able to inhibit the expression of key genes involved in the regulation of gluconeogenesis, and this is also and we are also able to show that this is decreasing the glucose production itself by the hepatocytes. And interestingly, so when you compare this effect to the effect of metformin, which is the most worldwide used anti-diabetic drugs, it's not as potent as metformin, and I can tell you that the mechanism of action is different, but at least we have the proof of concept that some antigen derived from these helminths can directly trigger metabolic tissue. Taken together, it seems that helminth infections or helminth-derived molecules can be beneficial in the context of metabolic disorders by improving whole body insulin sensitivity and also glucose homeostasis. So the question is, what I've shown to you is our studies done in rodents, and well, humans are not mouse, so it is relevant. Is it relevant to in the human context? So to answer these questions, I can show you some of the preliminary data we have generated on the field studies in Indonesia, in an island which is called Flores, and it's an island which is highly endemic for helminth. And some PhD students from the group, they have screened the population for helminth infections, and they have also collected plasma samples where they have measured glucose and insulin. And with these parameters in the fasting state, you can determine what is called the HOMA-ER, which is an index reflecting whole body insulin sensitivity. And what you can see on this slide is that people that are infected with helminths, and whatever the kind of soil transmitted helminths, they have a better HOMA-ER than the one that are not infected, suggesting that they have a better whole body insulin sensitivity. So at this stage, one of the questions, and I will end up my talk with this, is whether it's time for launching clinical trials with helminths for people suffering from metabolic disorders. Actually, there are more than 20 trials that were launched with helminthic therapies for some autoimmune disease. So it's safe. There are some that are under development and that might work. So the question is whether it can be envisaged in the futures for obese people, for example. So I want to thank you for your attention and acknowledging the PhD students and the postdoc that have done this work, Leonie and Noemi, and this project, this research line is driven in the group of Maya Yazdanbash in Leiden. Thank you for your attention.